Hello, my name is Trip Winans, and today I will be going over the CRISPR-Cas9 gene editing system. I'll give a little background on what it is, how it was discovered, and how it was revolutionized in 2012, and how we utilize it in gene editing today. So the history of CRISPR-Cas9 itself, so a little background of what CRISPR is. CRISPR is a term which means clustered, regularly interspaced short palindromic repeats. So I'll get to what that means in just a second, but it was not originally discovered as a whole. CRISPR DNA sequences was found in 1980 by a Spanish molecular biologist who studied at the University of Alicante in Spain, and he found CRISPR itself, but it was not revolutionized to how we use it today until about 2012. So the idea of the clustered, regularly interspaced short palindromic repeats, the most simply way I can put it is think of a library. You have bookshelves, and every single bookshelf is full of books. Now CRISPR, what it is think of for right now. We'll get to what it means sooner. Later as I continue to present. But think about every other bookshelf is empty, so there's spaces for new books to be put in. In this case, new DNA. So what is CRISPR-Cas9? CRISPR refers to the repeated DNA sequence genomes found within bacterium. So in other words, as you can say, as just how I said it was act as like a library, you can also picture it as like a memory storage card for bacteria. So in other words, those empty shelves I was describing about, think of that as a way in which when CRISPR cuts off segments of DNA, it can store them in there. So your next question probably is, oh, how can it cut off segments of DNA? Well, Cas9, that's attached in the name to CRISPR-Cas9, describes a protein 9 that is always bound to CRISPR, and that allows for the cutting of viral DNA infections. We'll get to it in a second, but in other words, it's a nucleonase, and anything that we know ends with ACE is usually an enzyme within the body. So CRISPR-Cas9 as a bacterial immune system. So we didn't realize that humans could utilize this until about 2012, like I said. So in 1980, the Spanish biologist discovered that archaean bacteria had utilized CRISPR-Cas9 against bacteriophages and viruses for many, many years. In other words, that if a bacteria is able to survive an immediate infection, it started to grow an innate immune defensive system, in which we call CRISPR-Cas9. So now let's get into it. There's three main stages to CRISPR-Cas9. The first one is adaptation. So upon infection, picture you have a cell, and a viral infectious DNA infects the cell. Well, at first, CRISPR, if it can survive the immune infection, doesn't die, it'll try to copy the DNA, correct? So, in other words, as soon as Cas9 cuts off segments of that viral DNA, CRISPR will go to work and start copying viral segments. And those segments that it's able to copy are stored in the CRISPR locus array. Now, remember how I said about the library and the empty shelves? Think of this new viral DNA that it just copied. It can store it. So now, inside of the cell, inside of CRISPR, inside of its own DNA, it now utilizes the viral DNA. And so now if that viral infection ever comes back, now it's like, oh, I've seen this before and I know how to attack it. Stage 2, biogenesis. So cRNA, otherwise known as CRISPR array segments and spacers, transcribed into long precursor RNA, processed into CRISPR RNA cRNA. So that whole idea of CRISPR copying DNA, it can transcribe it into RNA, and just for short term, it's just cRNA. Each cRNA contains the spacer stored viral DNA, like I said. And once that cRNA gets hybridized by a tracer RNA sequence, it becomes cRNA tracer RNA bound complex. Now, once you have this whole complex, it's recognized by the Cas9 protein. So think of it as in this viral DNA just got copied by CRISPR after it got cut off. And so now, once it's replicated in its own DNA, it has a whole RNA sequence called tracer RNA. And tracer RNA is going to find the cRNA and bind itself together and create this one singular complex. So once you have this one singular complex, now it can be recognized by Cas9, and Cas9 can bind to it giving the name CRISPR-Cas9. Stage 3, interference. So interference, Cas9 RNA complex patrols the cell. And so in other words, the Cas9 RNA is just a simplified term for the tracer RNA, cRNA complex. In other words, if you're getting a little confused, just Cas9 RNA is technically still CRISPR RNA. So think of it as like a patrolling police car. It just goes around until it realizes, oh, there's a viral infection somewhere. So at first, once it recognizes the viral infection, it realizes it must match the viral DNA spacer sequence. In the spacer sequence, don't forget, are the little gaps within the CRISPR DNA locus array. Okay? So, however, a short DNA motif called PAM, proto-spacer adjacent motif, must be present for binding. An example of PAM is NGG and Streptococcus pyogenes, which allow for the binding of Cas9. So Cas9 RNA is only able to bind to viral DNA if a short DNA motif called PAM is present. If the viral DNA does not have a PAM located or attached to it, then Cas9 is unable to bind to it. So once the Cas9 RNA binds to the base pair of its target, Cas9 nuclease, right? Remember I talked about how that's an enzyme, then cleaves onto the invading viral infection and destroys it. Think of it as like a Trojan horse. The viral infection does not know that it's what attached to its PAM receptor is Cas9. So in other words, CRISPR attaches to it, and then once it's attached, it releases Cas9 to destroy the bacteria. And how does it do that? Don't forget, because it had previously been infected, and Cas9 had already cut off snippets of it, and it had been stored in its own DNA, so then the cell recognizes it. So now we know how Cas9 works. Let's talk about how it's utilized in human gene editing. So, like I said earlier, the whole process of CRISPR was found in 1980, yet it is now revolutionized in 2012 by Jennifer Doudna, Emmanuel Charpentier, and some of their colleagues. Well, they had discovered that the dual components of cRNA and tracer RNA could be fused into a single guide RNA, sgRNA. And it's direct to Cas9, like they can directly link it to Cas9. And this is what launched the revolutionizing gene editing technology that scientists use every single day. So in other words, they were able to find a way to bind the RNA of the tracer RNA and the cRNA without having to go through a whole complex system. So the realm of gene editing. With that whole process in mind, there's two ways in which we utilize the whole process of gene editing. So we have a single strand RNA guides Cas9 to cut out any desired DNA location. So think of sgRNA as a guide for Cas9. It's just basically got it by the hand, walking it around until it realizes, oh, there's a viral infection DNA. Now Cas9, now go and infect it and cut off segments of the DNA. So once it does that, though, the cell itself also contains its own DNA repair mechanisms. Which allows for two main repair pathways that are utilized. One is non-homologous end joining that allows for the DNA ends to be glued back together. CRISPR can then delete or insert indwells, which is a nucleotide variation that allows for disruption in the gene. So think of it, there's a middle section cut out and it can glue it back together. Now the more common one is homology direct repair. This is the idea that DNA template is provided for the cell already, so the DNA already has like a whole set of instructions. And this allows for desired repair in the break. So once it's broken up, once Cas9 destroys it, it can correct a mutation by cutting it together and inserting whatever it wants to repair a mutation. And now that's important for a lot of disease-causing mutations within human cells or cancers. Basically, they can take a couple of the cells, cut out something they don't like, put it back together, and put in whatever segment of DNA they want to, what they think will work. So to conclude, CRISPR-Cas9 is a gene editing system that allows for cells to cut viral segments of DNA and store them into CRISPR locus library. So a cell contains CRISPR-Cas9. Cas9 can cut out segments of a viral DNA infection and use it almost like a template in a book. It can store it in itself. The cell then can recognize the viral infection if it ever comes back. And that was revolutionized gene editing technology in 2012 by discovery of sgRNA, the single RNA strand. And that allowed for two main types of gene editing just talked about, non-homologous end joining and homologous direct repair. Thank you.